Good afternoon, and thank you for coming to listen to my presentation today. I'm sure the post-lunch food coma is well on its way, but I appreciate your time. And I apologize for not being there in person, and I'm very grateful for the opportunity to still present to you by means of an audio recording. So thank you very much to Nadine and to the coordinators of the Ocean Sciences meeting for allowing me to do this. Today I'll be presenting results of a study on the effect of autumn and winter diet on the physiology of juvenile Antarctic krill. This work could not have been done without a number of people involved, but especially my co-authors, Kirsten Steinke, who's currently a PhD student, and Julia Fontana, who was actually an undergraduate at the time of the research and graduated with her ocean sciences degree while we were in Antarctica last austral winter. And just because I'm not in front of you and it's useful to have a face to put to a voice, this is me from Antarctica looking like someone who's just spent 6 months in the winter in Antarctica. Okay, so Antarctic krill are an extremely abundant and highly successful zooplankton species that plays a major role in the Southern Ocean as an agent for nutrient and elemental cycling, a key prey item for numerous top predators, and the focus of the largest Southern Ocean fishery by tonnage. The success of the Antarctic krill population relies on the ability of young krill to survive the long, dark Antarctic winter, where food is extremely scarce. But how do they do this? Well, we think the key lies in their autumn diet. Antarctic krill have a wide range of dietary options available to them, from herbivorous through to carnivorous. They are known to feed on diatoms and sea ice algae, but also other zooplankton such as copepods and protozoans. Diatoms provide krill with polyunsaturated fatty acids, or PUFAs, which are used by the krill for growth. Zooplankton, on the other hand, provide krill with triacylglycerols, or TAGs, and these are used by krill for energy reserves. So the question is it better for young krill to grow by feeding on diatoms in the autumn in preparation for winter starvation, or is it better for them to store energy reserves by feeding on copepods or other zooplankton in autumn in preparation for winter starvation? And if they can find food in the winter, what type of food is best? Diatoms or other zooplankton? Now you might be wondering why this is important to know. Surely they can just find whichever food source is best for them and eat that. Well, long-term studies along the Western Antarctic Peninsula have shown that summer diatom biomass has decreased in the last 25 years and is projected to continue decreasing with continued warming and sea ice decline. During that same time period, summer copepod abundances have risen, also along the Western Antarctic Peninsula. So the food available to Antarctic krill in this region in particular is changing. So with all of this in mind, the overarching objective of our project was to experimentally determine the effect of diatoms versus copepods, on the health of young krill in autumn and winter. Our study involved a long-term feeding experiment in which roughly 700 individual juvenile krill were placed into 4 2,000-litre circular aquarium tanks. So that's 700 in each tank. We collected the krill on the southbound voyage at night in the Bransfield Strait and in Wilhelmina Bay. I won't go into too much detail on the experimental setup, as I'd really like to focus more on the results, but at the end of the presentation you will find my email address and my Twitter handle, and really please feel free to reach out to me and ask me any questions you may have, and I'll do my best to get back to you with some good answers. So briefly, our experiment was divided into 2 time periods. Period 1 covered June and July, and period 2 covered August and September. Half of the krill were fed a freeze-dried and ground mixed zooplankton assemblage, which I will refer to as MZA. This zooplankton assemblage is one that we collected ourselves off Palmer Station. The other half were fed a live diatom culture, also originally started at Palmer Station, and I'll refer to this as dia. The dominant diatom in the culture was a Cynodropsis species. So during our experiment, krill were either fed consistently through both time periods, and these treatments are illustrated in the experimental design diagram here as the bottom two circles. I will refer to these either as MZA or dia. Or they were only fed in the first time period, June and July, and then provided with just natural seawater for the second time period, August-September. So this group were essentially starved. And I will refer to them as either MZA nat or dia nat. At set time points, every 4 weeks, starting with an initial time point in May, we removed a subset of krill from each tank and ran a series of experiments on them, including respiration, ingestion, and egestion rates. We also collected krill for measurements of lipid classes, fatty acids, protein content, carbon and nitrogen content, and reproductive development. All krill were measured for length and wet and/or dry weight. I will only be presenting results from our respiration rate measurements and for condition factor. Condition factor is a metric for the health of an organism and is typically considered as the ratio of dry weight to cubic length. Because we don't yet have our samples back from the field season, we haven't been able to analyze and process our lipid samples, and so condition factor is really the best thing that we can use right now. So here we have mean condition factor for krill from the MZA and MZA nat treatments on the left, and the same for those krill in the dia and dia nat treatments on the right. Month is along the x-axis, with May representing the initial time point and time points 1 through 4 being conducted each of the subsequent months. To start, you can see that the condition factor immediately and significantly declined for krill in all treatments. The condition factor continued to drop into July in both treatments, but significantly more so in those krill fed the diatom diet than those fed the MZA diet. After our July time point, the treatments split into those with continued food supply and those with only natural seawater supply. At this point, condition factor declined further, with those krill on the nat diets having significantly lower condition factor than those krill with continued food supply. By the end of our experiment in September, we noticed something quite surprising. The condition factor of the krill in the MZA treatment increased significantly. In contrast, condition factor of krill in all the other treatments continued to decline, with the lowest values being observed for krill in the dia nat treatment. So what can we take from this? Well, condition factor clearly declines when krill are starved. I don't think that would be too surprising. But a carnivorous diet improves their condition factor. And a carnivorous diet throughout the winter seems to allow for an earlier recovery of condition factor. This slide layout is the same as the previous one, but instead of condition factor, we have respiration rates. As you can see, respiration rates did not vary significantly between our initial time point and our first time point in June. But respiration rates increased significantly between June and July, most likely in response to being fed. But this is an interesting finding because adults in other studies have not responded to being fed and have maintained their very low respiration rates. By August, after our treatment split to half on continued food supply and half on natural seawater supply, we found that respiration rates of krill that started off on a carnivorous diet were not significantly different from those that were fed the carnivorous diet for the duration of the experiment. In contrast, we found the opposite in those krill fed the diatom. So those krill that were started off on the diatoms and then starved showed a significant drop in their respiration rates compared to those krill that continued to be fed diatoms. We suspect that this is likely the result of their limited lipid storage. I really expected that these results would continue, or these trends would continue the way they were going, and was very surprised with what happened next. So between August and September, respiration rates of krill in the MZA nat, and dia treatments declined, while those in the dia nat treatment remained relatively stable. This is quite surprising for the krill in the MZA and Dia treatments because they were being fed throughout this period. Also, the krill that were in the MZA NAT and Dia treatments had increased food availability from what they had experienced in August and July and June. This is because the early spring bloom had begun. So the fact that the respiration rates actually went down in 3 of the treatments and kind of remained stable in one of them was not what we were expecting. What can we say from this? Well, a carnivorous diet in autumn prepares krill for later starvation in winter. Respiration rate decreases in late winter, even if they're fed, and we're not really sure why. So what are the implications of our findings so far? Well, it seems very clear that a carnivorous diet is really important for juvenile Antarctic krill during the autumn and, if possible, during the winter. But it certainly seems to stand them in a better stead than if they fed on diatoms alone or started off on diatoms and then were starved later. So the increased abundance of copepods that we've seen along the Western Antarctic Peninsula in the last 25 years seems to be advantageous to overwintering juvenile krill. This is great. What does that mean about the decreasing abundance of diatoms, though? Does this mean that there's no effect of that? Well, I don't think we can jump to that conclusion, because we do know that reproductive development and growth in krill relies on diatom consumption. And some of our other samples and data that we were collecting during the winter and during this long-term feeding experiment will help us address this aspect of the question in greater detail in the coming months. I'd like to acknowledge the entire Winter Over team from the Palmer Station winter of 2019. Without their incredible support, we really would not have been able to do what we did. The research was funded by the National Science Foundation, Office for Polar Programs, and I'm grateful to their support. I'm also grateful for the support of the United States Antarctic Program. Thank you for your time.